And that's a little bit worrisome because Americans are interested in getting well with a pill. Now, it could either be a pill or it could be a metaphorical pill. In fact, in the American culture, one fad after the other comes along and people grab onto that in order to get well. The current fad, you'll probably all leave when I tell you what the current fad is, but the current fad is, well, it was low carb. Do you remember that? That was a fad. You may not know this, but that is a completely, not only unsubstantiated, but a crazy idea that is poor nutritionally. But that was a fad. And so all the people that manufacture stuff, they know that if they put on their low carb that you might buy it. I mean, it might have been low carb before. That doesn't matter to them. They'll put it on there now, see. The current fad is to avoid wheat. That's the current fad. Now, here's the problem. What's coming out of this genetics world is people are going to want to get their genome examined and have somebody fix them. And the way to prevent cancer, folks, is not by fixing your genes. The way to prevent cancer is to change the way people live. But that's hard work, and we don't like to do it. We don't like to do it, and we don't like nobody to tell us to do it. Aren't we kind of like that? Yeah. We actually think you might prevent 90% of cancer by changing the way you live, irrespective of genetic issues. So maybe that's enough on that. Why something I know what the question is. Why something stem cell research? Why embryonic stem cell research if from any cell we can reproduce any part of the body? Now, if you weren't here last night, and you will hear tomorrow evening, you'll hear Dr. Carson speak of this. We can take a cell and we can, this is a metaphor, back it up. And then it becomes a stem cell, essentially, and we can redirect it to become some organ in the body. Remember that from last night? That's what this question is about. And the person is saying, why go after embryonic stem cells? And I thought I mentioned last night that the reason we don't have to do that is because we can take regular cells and make them into, effectively, stem cells. Did you all follow that idea? And in case it needs a little more explanation, we can take a stem cell, put it in a plate, and depending on what we feed it, we can make it start to become a kidney or a nerve cell or whatever. We've learned to do that. It's amazing, wouldn't you say? Just one cell. That's the way the baby was made. As that little trophoblast is starting to multiply, there aren't a bunch of cells that to get together and say, let's make the heart. One cell says, I'm going to make the heart. You all with me on that? One cell says, I'm going to make the brain. Now, they're all talking to each other. It's astonishing, folks, beyond imagination. And we'll get to some more of that later. So, interesting. Good question. Very good question. What is the percentage for men in getting breast cancer? About 1/100th the rate of females. Now, interestingly enough, I don't know if I should do this, but here we go. Inside the female breast, there are these little things we call lobules that produce milk. And together, they put their milk in a duct. A duct. And 82% of breast cancer occurs in the lining of these ducts. Y'all with me? And the reason that happens is every cycle in the female menstrual cycle, all of these cells die in that lining, and before they die, just before they die, they copy themselves and make a new lining. So you get copy error. Are you all with me? And you get a place where there is a high likelihood of, because of copy error, and that's where 82% of the cancer occurs. About 12% of the cancer occurs in the linings of the lobules for the same reason. Now, you may not know this, but most of you have some idea of this. But when a little fetus, and I can't tell you which day it is, but you'll see this on the fourth night. What would the fourth night be? Is that going to be Wednesday? Fabulous 15-minute clip of the development from fertilization to birth of MRI imaging of the whole thing. Just a fabulous piece. And there is a place during that development when the sex has not yet been determined, but structures like breasts are developing, and men are actually born with some that's a word scientists like to use, not fully developed, but some proto-structures just like this. Are you gals aware of that? Have you ever heard that that is in the literature that there are some men that have been able to nurse? Did you know that? To lactate. That's in the literature. And it's because before the sex was determined, certain things are built there already. So when men get breast cancer, it's also usually in one of those places, although there's no cycling going on. So it's an interesting story. That was all free. You didn't pay for that. I just thought you'd be interested. How does the mapped genome differ in each person? This is a very good question, which I'm going to set that aside because we're going to take our knowledge of DNA tonight just a little bit further. Do you think you could stand that? Say yes. All right. You showed us what the genome looks like. How does the gene portion look different? Coming up. I thought lysine helped to stop cold sores. I've never seen a study that showed that. I know that people believe that. And by the way, this is another problem. If you give people sand, 53% of them get better. Did you know that? What do we call that? The placebo effect. So I've never seen a study. It may be true. It doesn't matter. The point is you get enough lysine if you just eat. You don't have to buy it in a bottle. You can if you want. And if you took some, you'd probably feel better because of the what effect? Because of the placebo effect. Did you know a study was published less than a year ago that showed that even when people knew they were getting a placebo, they got better? Did you know that? Did you see that? Yeah. Now this is a tougher one. What are your thoughts on Richard Dawkins' book, The Selfish Gene? Is this a confirmation of God's command to be fruitful and multiply? Or does this concept even exist? We need more time than I have on this, but Dawkins wrote a book called The Selfish Gene in which he was trying to, he built a theory that said if there was something about the organism that could do better, the gene that would make that happen would somehow be preserved. So he said it's like the gene was selfish and it's going to make itself, it's going to make sure it got preserved. You get the idea there? That's what's in the book. And actually when he wrote the book, even as it was being published, he wrote himself, it's not the best name for the book, The Selfish Gene. The point is that he has this theory, it's not been shown to be true, that somehow because that gene, if it could be expressed, would make this organism better and then would be able to survive and so forth, that gene would be protected in some manner. Maybe somehow even protected it by itself. But that's not been shown to be the case, but that's what that book is about. And I don't think that's part of what God's plan was when he said be fruitful and multiply. That would just be my thinking. Somebody asked me if I ever worked with a guy named Joe Cruz. The answer is yes, for 5 years, many years ago now. And there's an interesting question here on the Bible, which I think I'm going to leave aside for the moment. Last one, if you have this BRCA mutation, is there any way to change it? You understand that the BRCA gene is good for us because it makes a protein. And by the way, watch this point. This is new tonight. Genes have you know this, they have the code or the information for the cell to make some protein. And it's the protein that does the job. Whatever the job is, the protein causes it to happen. This is an amazing thing, folks. How the proteins that cells make go out and make things happen in our bodies, see. all kinds of things. And so if the gene that this represents, which is a section of ladder, you all with me on that, I think, aren't you? And I don't know how long the section is. It wouldn't be hard to look up. But if there are some mutations that do not cause a problem in terms of its ability to suppress cancer, there are just, and what shall I do? Shall I put an X there? That one wouldn't be, that mutation wouldn't be a problem. It's not that the rung got lost, it got changed. But there are other mutations, and I'll put a circle there, that if that's the mutation that you have, then this protein will no longer be able to do its job of suppressing tumors. Did you follow all of that? And when that gene is inherited, every cell in my body has that gene. All of us have that gene. We have 2 copies of it, one from mom and one from dad. On the chromosome, one chromosome from mom has that gene, and my chromosome from dad has that gene. And one or the other could be mutated, and if it's the right mutation, then I've got this problem. And yes, it's in every cell of my body. There's no way we know of to fix that. If you can actually take one cell and play with the DNA, but there's no way to do that with a whole bunch of cells. All right. I wanted to show you some more pictures of grandkids, but I thought I'd spare you. Tonight is the Behe interview, and in just a little bit, tomorrow night it will be the Carson interview, and Wednesday another one, and Thursday another one. We'll do questions each evening, and actually this lecture series is available. The problem is I do it different every time, and so the one you bought would not sound exactly like this one, but it's still, I didn't know that was there. I honestly didn't. You'll never believe that, but that's actually the truth. Anyway, just a little quick review. What's in the middle of this page? That's the DNA or the genome, a piece of it anyway, isn't it? And for those of you that weren't here, it's like a ladder that's twisted in opposite directions, and these rungs are actually half rungs, and the rungs themselves end up being the code or the information that allows the cell to use that information and make a protein. In 1953, most of you are familiar with this, 2 scientists by the name of Jim Watson and Francis Crick discovered the structure. They discovered that it was a ladder of DNA. You can't tell it very well, but in their laboratory with these clamps and these bars, they actually formed the molecules and hung them in midair, and you can't see it real well, but can you see that angle right there? That's one side of the helix, and there's the other one going the other way. Now, Crick's wife, Odine, was an artist. And notice this picture, and we'll come back to that. She passed away a couple of years after he did. He died, I think, in 2004. But anyway, Odile, I'm sorry, I said Odine, is an artist, and so she drew this for them. And this has become a very famous picture. It's in almost any biology text. It's become kind of a, I think I even put it on here. No, I didn't. But it's a representation of this wonderful discovery they made, and you all know they got the Nobel Prize for that. All right. So a quick review is, it's a ladder. It has over 3 billion steps or rungs, and it has our genes and a large portion of non-coding rungs. Do you remember what that means? A whole lot of the DNA does not code for proteins, right? And Francis Collins called it junk DNA, to his shame, we know today. But nevertheless, and this is something I didn't mention last night. You're going to hear this if you do much reading. These little half rungs, y'all with me on what I'm talking about, are actually alkaline chemicals. There's 4 of them. They have names, and then the names start with A, T, and C, and G, correct? And In the world of chemistry, we have a different word we use when something is alkaline. We say it's basic, acidic and basic. And so these are base pairs. Got it? That's the way the scientist refers to them, as base pairs. He doesn't call them rungs in the ladder. We did that to help us a little bit to visualize, right? Okay. Even if the rungs were only spaced apart by a thickness of paper, the ladder would stretch over 200 miles. All you got to do is take the thickness of paper and divide it in, and you'll find out how many miles that would be with 3,200,000,000 pieces of paper. There's a kind of a drawing, untwisted, that I've made, and I've colored the T's and the A's and so forth. You probably know this name D, or these letters DNA are exactly the name of a chemical. Most of you have heard deoxyribonucleic acid. Don't worry about that, but you've heard that. It's just the name of the chemical. The length of this ladder, because those rungs are 1,000,000 times closer than a piece of paper, in the cell, the length of the ladder is over 3 feet, 4 or 5 feet perhaps. And these rungs are close. There's not much space between them. And I'm going to show you a clip done by Harvard that simulates, computer simulation of how this 4 or 5 foot thing gets folded up into about the space of 1/1,000,000th of a meter, a micron. And actually it's about 1/10 of that. So let's go over here and find out where that is. There we go. Let's make it big. I'm going to stop it. Can you see the twisted ladder? Everybody talk to me. Can you see it? And the twisted ladder is first of all wound around some proteins called histones. So you could say it's wound, and then it's coiled like so. And then the coiled piece is folded by another protein, and then the folded chain is coiled once more. Can you see it being coiled? And on the right hand side of the screen, at least at the magnification, is kind of a large tube. Are you all with me on what I'm trying to describe? And that large tube actually is what we see as the chromosome. You'll see this in a moment. The simulation is trying to show you that those large tubes became the pipes that make the chromosomes. Y'all with me on that? So we have wound, coiled, folded, coiled, and this 3, 4, 5 foot thing becomes like that. Got it? It is amazing, is it not? So there's the double helix, there's the histone proteins, and then I think you got this. This is that large coil, correct? I wish you'd all talk to me when I ask you a question, but and there's the other one. One of these is from my father. I spoke incorrectly. I said it was a half ladder. It's not. The gamete, that's a fancy word for the sperm or the egg. It would refer to either one. The gamete has half a set of chromosomes. It has 23 chromosome halves. You all with me on this? And I think I said half ladder, which would have been, could have misled you. So we get 23 chromosomes, but each chromosome is 1/2 of the chromosome came from my father, 1/2 came from my mother, and they're buttoned together as we have talked about before. Now, what does it do? This is a quick summary, and we'll add a few extra things. Every night we want to expand your knowledge of DNA just a little more. It provides a pattern for making proteins. Here's the new point. The rest of the DNA We now know, even though Francis Collins thought it was junk, we now know that it has a controlling feature over whether the genes get expressed or not. I want you to learn what I mean by that. Expressing a gene means the cell used the pattern to make a protein. So if the gene was repressed from being expressed, what would happen? That particular protein would not be made. Right? And so we have learned just in the last several years, and if you make the connection with the ENCODE project from last evening, did I tell you that there were 400 scientists that had been hired from around the world, and they are studying that one question: what does the non-coding portion of the DNA do? And they have made amazing progress, and that's the field of epigenetics. Anybody heard that term before? You've all heard it now. What does epi mean? It means beyond or above. And so it's just a name that they thought that would help describe this thing. And this controlling stuff, folks, is becoming huge. We already see that it has huge implications in our body's functioning, in health, etc. It's just an amazing story that's developing out of the so-called junk DNA. Isn't that amazing? Are y'all kind of clear on what I just described? Epigenetics, this field of study, how the genes are controlled. And it has amazing implications. Okay. So while the blueprint looks something like the final product, the DNA uses a code made of these 4 alphabetic letters, which you already know. These are the first letters of the 4 bases, or the 4 alkalins, or the 4 half rungs. Correct? The 4 half rungs that are just used over and over and over. All 3,200,000,000 half rungs are either A's or. And I showed you last night that example. I didn't show you that A's are always connected to T's, and that C's are always connected to G's. And this is why the artist who drew this picture made only 4 colors, and he always matches a greenish color with a what? Kind of a reddish color. Y'all with me? Anywhere you look on here, he's done that. Is that green with red? Or anywhere he has a red, he matches it with a, got it? Watson and Crick are the ones that discovered this. I'd love to tell you how that happened, but we don't have time to do all kinds of things. And the G's are always hooked to C's, and vice versa. C's are always hooked to G's, and so forth. Now, well, of course, they wanted to map the whole thing, and that's, we'll see that again in just a second. Just like, friends, this is just a simple example, but it makes sense, where the word tile and light have the very same letters, but the order of the letters makes a great difference, doesn't it? And the same thing is true in the genetic code. In fact, that's what we call it. We call it the genetic code. Even the portions that are non-coding, still with me? We still call that genetic code, because those codes are going to have something to do with controlling the expression of genes. Is that correct? Whether the gene gets used to make a protein, which is going to go off and control something or do something, see? In our DNA, the code is, this is new, this is another new thing tonight. The DNA, and I'll show you this in just a moment with a simulation, a computer simulation. The DNA is designed that 3 letters in a row code for, are you with me, an amino acid. That kid's already getting it, isn't he? None of the rest of you said anything. So ATT is different than ATG. ATT codes for a certain amino acid, and ATG codes for a different amino acid. See, and Watson and Crick, they learned all of this kind of at once. They've been working on it, of course, for several years. And they got it all put together. Quite remarkable. All right? What do? They have the instructions for the sequence of amino acids then, for building proteins. And these instructions are used by a protein manufacturing factory to assemble the protein. And always, no exception, this is always done inside the cell. And in the picture I put on the screen last night of the cell, what symbol, what little mark on that drawing was representing a protein manufacturing factory? Do you remember? The little black dot. Now they have names. The names are not that important. But some of you have heard this word. It's a ribosome. That's the name of the factory. The black dots. And always proteins are manufactured inside our cells. Now this is free. This is like a nutrition lecture for 30 seconds. We don't use any proteins that we eat as such. Our body takes them apart, sends the amino acids to all the cells in our bodies, and our own bodies make all the proteins we need when and where we need them. Did you get that? Widely misunderstood by the American public. Widely misunderstood. All right. So what are proteins? You know this. They're a string of amino acids hooked together. The factory hooks the right ones together in the right order in this long chain. It's then folded up into a special shape because proteins work because of their, everybody should say it. All right. Let's watch Harvard's simulation of this working. And here I have one more slide, just simply illustrating that 3 rungs at a time, or half rungs if you wish, are used to code for a certain amino acid. And let me give you, I forgot I was going to show you in a simpler drawing of a little bit how this works. Here's the ladder, and there's a little machine that comes along and opens the ladder up like this. Remember the rungs are not quite hooked together, sort of? They're hooked, but hooked with a kind of a easily broken connection. And then another little machine comes in and makes a copy. Watch this. Watch my arrow. It puts an A here where this A used to be, and a G here where this G used to be, and a C where this one was. You up with me? There's one little trick here. Whenever there's an A, instead of putting a T, what does it put? A U. What's that about? Once this thing is made, it is removed and taken to the factory, which reads the code and builds the protein. Y'all with me? Yeah. And because of this little feature that I didn't tell you about last night, we call this RNA. I know you know the term. You've heard of RNA. Have you ever heard the fact that the HIV virus is a retrovirus? Have you heard that term? A bunch of you have. That's because viruses are normally made of DNA. But the HIV virus is among a class of viruses that are made from RNA. And so we call them retroviruses. Just so you'll know what you're talking about or what you hear. Okay, so, and then here's a chain. This artist, this is out of a textbook I have. The artist in the textbook has used triangles to represent amino acids. I was using little balls last night. You could use anything you wanted to represent the different amino acids. Y'all with me? And it's kind of folded up here. All right. So here's what's going on. In a moment we're going to see the computer simulation where guess what this is? That's the RNA that's inside the factory. The factory's reading 3 half rungs at a time. Are y'all with me? To decide which amino acid to hook up next. And it calls for a truck that carries the amino acid. And it's in the back of the truck. And on the nose of the truck, this is the truck. This is the amino acid. On the nose of the truck are 3 half rungs. corresponding to this amino acid. So you got to have a different truck to haul each different amino acid. And this is how the factory can make sure that it assembles them in the right order. It only allows a truck to come in with the half rungs that match the first 3 half rungs of the RNA. Isn't this exciting? Okay, let's watch. I'm going to stop it for a second and make it go mostly full screen. We're inside the cell, looking at and moving toward the nucleus, which has these little pores, so stuff can get inside and back out. And the pores have a sentry, interestingly enough, that you'll see in a moment. So the camera right now is just going inside the nucleus, and momentarily, when we get inside, you'll see, guess what? What do you see? You see the DNA, you see the twisted ladder, you see some other stuff, of course, too. And the computer simulation brings us close. Now, this is an oversimplification because this DNA has been coiled, wound, coiled, folded, and coiled, hasn't it? But so they're showing us a simpler form. And actually what happens is it's partly undone so that these machines can get in and there do their work, which is a whole nother fascinating story. But right now there's a machine coming along which is unhooking the half rungs. Can you see it? Then another machine comes along. It's got a name, of course. All these things, we've given them names, which are not terribly important. And that machine is building the half-rung copy we are calling now RNA. Is that correct? And then that thing is also disconnected, and it's ushered by a machine that they don't show. There's a machine that's carrying this. And when the RNA gets to the pore, the sentry says, wait a minute. Oh, okay, I recognize you. I'll let you through. You see that? This is going on, folks, in every cell in your body. And it is carried along a highway that they don't show. It's actually a molecular highway, and taken to the factory, which is in 2 pieces until the RNA is laid in there, and the pieces are hooked together by another machine. And then inside the factory, it starts to, watch this now, build the what? The protein. Y'all with me? Even though you couldn't say it, now you know it, right? Once I said it, you said, oh, that's right. Yeah, that's right. It's the protein. All right. Now watch him, what he does. Watch what the truck does. He moves up and matches himself. Uh-oh. He doesn't want to do it here. I'm sorry about this. There he goes. He matches himself. And if he fits, he doesn't move. Watch this. He waits till the next truck arrives, who fits the next pair, or triplet, and then he can leave because his amino acid gets hooked up to this one. Got it? Then comes the next truck. This one can now leave, etc. Got it? Is this cool, folks, or not? Does this thing look like somebody planned this, or did this just kind of happen by chance? And I don't want you to laugh, folks. If somebody has the opinion that this happened by chance, let's not laugh at their opinion. Are you all with me on this? But let's recognize the hand of a designer.
Is there ever a mistake?
Yes. This thing happens so fast, with blinding speed. Like in a few seconds, you can make a long chain and fold it up. It's just astonishing. And once in a while, there's a mistake. But since you asked, The body has a whole system of peptidases that travel around your body looking for proteins that shouldn't be there, and they dissolve them up into amino acids. Phew. I hope I have time one night here to play for you a clip. I'm sure hardly anybody's ever heard of this guy. His name is David Berlinski. Anybody? You've all heard it now. This guy really fascinates me. He is so articulate. I just roll my eyes when he's talking. I talk like kids. I'm sorry, not all kids are inarticulate, but and he is not a believer. He is absolutely clear that it was designed. He just doesn't know who did it. Now that's not such a bad place to be, is it? I don't know who did it, but it's been designed. There's no question about that, and it's fun, it's wonderful, I think, to hear him talk about that. So we'll see if we can squeeze that in. But I have a couple of his books. I can read about a page or two at a time, and I got to go sit and think for a while. He's a genius. And by the way, from his name, what do you suppose his national background is? He's Jewish. God gave those people smart brains, folks, that the most of us don't have. I'm telling you, it's just the way it is. Okay, so this is just going on until we get 100 or 200 or 300 or 400 or 500 amino acids hooked together. And now they're showing you the simulation from outside. What's this chain down here? That's the protein, isn't it? What's this chain up here? That's the RNA, isn't it? That's the pattern. Okay, good. And what's this thing? That's the factory, right? Okay. And now, they don't show this, but there's another machine that takes this chain over to a folding factory. And it takes it inside the folding factory, which, by the way, is made of protein. The protein manufacturing factory is made of protein, and the folding factory is made of protein. Are y'all still with me? And listen to this. Inside the folding factory, there are some proteins we call chaperones that fold the protein up into the right shape. Are y'all still with me? Does this sound like this was designed? It's mind-boggling. Now, they don't show the chaperones, but they show the protein folding up, which even one that's several hundred amino acids long is folded in just a couple of seconds. It's just astonishing. And you understand, nobody has ever seen this. This stuff is too small to see. So here you have this huge problem in scientific research, folks. How do you figure out how stuff works that you can't see? That's a pretty interesting question, isn't it? And it's fun to talk about that and learn about it. Nevertheless, it's all folded up now, so the factory is opened. And finally, they do show the protein truck that picks this up and takes it where it's supposed to go. Got it? Now, I want you to hear a fellow named Dean Kenyon talk about this.
Perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture.
I'm going to tell you more about this fellow, Dean Kenyon. He was once one of the most famous atheists in the world. Interesting story. Wrote a book. I'll tell you about that in just a moment. And you can probably tell by the look on his face that he has become a believer in design. Are y'all with me on that? He's a professor in a university. He's just blown away by this.
And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on...
Where we see what? And then I interrupted him, but he says, on Earth.
On the Earth.
and so forth. All right. As Anthony Flew said, inordinately greater integrated complexity that we have learned about only in the last 10 years, 10 to 15 years. The plot thickens, however. Dean Kenyon, the man I was showing you a moment ago from San Francisco State, a typical evolutionary biologist who formed a theory on how amino acids could hook up together in the right sequence, and I didn't put this in there, by themselves, based on inherent, as it were, or built-in information. The name of his book was Biochemical Predestination, and watch what it says. I wrote this down. He co-authored this book, and this book was the source of great hope and anticipation in the worldwide evolutionary community, because they believed that they had found how proteins could, by themselves, form the structures that were needed. Are you all with me on this? This book was heralded the world over not many years ago. And respected everywhere. But notice what he says.
It was during that whole period of time that my doubts about certain aspects of the evolutionary account became apparent. When coming into contact with a powerful counter-argument given to me by one of my students, and I could not refute that counter-argument.
Now, the student didn't come up with the idea. The student was reading a book and recognized a connection with what the student was reading in the book and what Professor Kenyon was teaching, and brought it up to him. And Kenyon read the book, and he said, and this is while his own book was in its maximum interest the world over. He's saying, it was while this was happening that I started having these doubts. And he read this, and he realized that he needed to go back to his laboratory, and I have another clip of him saying, I think it's on the next page, here it is.
The more I conducted my own studies, including a period of time at NASA Ames Research Center, the more it became apparent that there were multiple difficulties with the chemical evolution account. And further experimental work showed that amino acids do not have the ability to order themselves into any biologically meaningful sequences.
Did you get that? Remarkable. And he is now one of the leaders in the intelligent design community, having written this book that the world thought, this is going to be our answer. This is going to show us that evolution could somehow work because the proteins could hook together by themselves. Well, here's Michael Behe, and let's do the interview now. I'll let you just hear one thing that he says, and then my interview with him.
When we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
All right. I think I have it over here. And is this the right one? I don't think it is. Sorry about that. Yeah, wrong one. So let's try again. I thought I had it set up ahead of time, but it won't take us but a moment to find it. Design or evolution, or some people would say creation or evolution. And in a certain sense, Dr. Behe, there are people likely that believe that there was design, but maybe they would say, I don't really know how it happened after that, but it's obvious to me that this thing was designed.
Yes, that's right. And that's the nice thing about design, because you can come to a conclusion of the design just by looking at a system or something that's in front of your eyes and saying, it might have been created ex nihilo, or somebody might have put it together,
piece by piece.
Piece by piece. But however it got here, it was intentionally made. It's not an accident.
It's too elegant.
Too elegant, too functional, too complex.
Now, in spite of the fact that you were raised in a Christian home, attended parochial schools, you were taught, even in the parochial schools, you were taught evolution. Give the story of how you came to embrace it, believe it, and then let's trickle over into how questions started arising in your mind about it.
Well, yes, I went to parochial schools when I was in grade school and high school, and whenever we talked about how life began and so on, well, we always realized that God was behind it, but the question is, how was he behind it? And the idea was that we were taught was that God made the universe and its laws and knew how it would unfold, and if he wanted to make life by secondary causes, then who were we to tell him that he couldn't?
Kind of a theistic evolution.
Yeah.
And there are many varieties of theistic evolution, of
there
course.
Are.
But,
yeah, there are. But that sounded fine to me. I didn't, that was not my main interest. And then throughout high school and through college years and graduate school, you know, that was my position. You know, God made life, but Darwin's theory of evolution, as I learned in grade school and high school and college and so on, was the mechanism by which life arose. And it wasn't until later that I came to change my mind about that.
Before you get to that point, all through graduate school, there was really no room for anything but evolutionary mechanisms in the teaching?
That's correct. As a matter of fact, in graduate school, we weren't taught evolution. It was assumed. You know, this arose through evolutionary processes.
Yeah.
But if, you know, when you study biochemistry, which I studied in graduate school, you study enormously elegant and complicated systems that keep your cells and organs and so on running. And it was enough just to try and learn it. That was enough effort.
Don't try to think about how it got to be, right?
But yeah, we were not taught, you know, this is how it arose, or this is step by step how a random change came about in this enzyme or this piece of DNA or so on, and it was an improvement and led to this particular system.
But surely there must be times in most of these professors' thinking where do they ever ponder this kind of thing, or did they just keep assuming?
A lot of them simply assume that's the way the world works. And I've met a lot of people who are like me, that is theistic evolutionists, who will say that's the way God should make the world. That is, that
start it up and leave it alone.
Start it up and leave it alone, because if he doesn't, it violates its integrity or some such thing.
Yeah.
Rarely does one get past a certain age and then reconsider one's beliefs in
the underpinnings that started it all.
That's correct.
Well, then tell us the story of how this started to be a challenge for you in terms of what you were so clear that this is what had happened.
Yeah. I should emphasize that I was completely fine with it, and I used to enjoy making fun of people who didn't believe in evolution. You see all the people like Richard Dawkins and so on, and these days, but you know, I did the same thing. It's the nice thing about believing in evolution. It automatically makes you smarter than anybody who doesn't. I was a postdoc and then became a professor at Queens College and City University in New York, and then I came to Lehigh, and I saw an advertisement for a book called Evolution, A Theory in Crisis by a man named Michael Denton, and he was a geneticist, an Australian geneticist. And I was intrigued because I had never...
Even considered?
Well, never considered. I never heard of any practicing scientist question Darwin's theory.
And he had credentials?
He did. He was a medical doctor, a PhD, a geneticist who studied eye diseases, and had given a lot of thought to this question. He was also an agnostic, too. He didn't have a religious goal to pursue. He was just tired of hearing Darwinists claim a lot for their theory when he saw very many problems for it. And so through his book, he very fairly went through and explained Darwin's theory, why it was attractive, what it can explain, and then went devastatingly and said, well, but look at these things that it doesn't explain. And
this is a really interesting and compelling section of this interview. And I'm going to, tomorrow night, a few paragraphs from his book, show you some of the things he says. What did Michael Behe say about the philosophical position of Michael Denton? He was a what? He was an agnostic. Interesting. Now, he has changed his opinion, by the way, and that's an interesting story in itself. And follow carefully this pathway for the next 2 or 3 people that Michael Behe tells the story of how this thing was developing. And I'll put pictures on the screen of each of them just to help give some context. And we'll hear from them to some degree each evening later on.
And by the end of the book, I was astounded because, you know, I thought back and I realized I believed in Darwinian evolution. I asked myself why, and I couldn't remember. I didn't. You know.
You didn't know why.
There was not a reason. It was because that's what everybody believed. You know, you can say a few things. This looked like that, and so on. But, you know, getting from one place to another is more complicated than one thought.
Especially in the last 15 years.
Oh yeah, absolutely. But even back in the day, in the late 70s, early 80s. Well, ever since people built up the science enough to realize what was the foundation of life, that molecules were running the show.
Yes.
It's like the nuts and the bolts in your car. You know, the hood and the exterior might look real nice and attractive, but those nuts and bolts better have the right shape and the right strength and be attached to the right other nuts and bolts and so on to make the car work. Ever since biochemistry began to be able to study those, things have gotten increasingly complicated, and that has not stopped. But by the time I was in graduate school and then early years as a professor, it was known that many things were outrageously complex. And after I read Michael Denton's book, I said, well, I had assumed that all those things had evolved by Darwin's mechanism, that is, by random changes in the natural selection.
Natural selection.
But if somebody has explained it, the nice thing about science is that they would publish it in the scientific journals. And the scientific journals are in the library.
That's the rule of life in the scientific community.
Publish or perish.
That's right.
So I went over to the library and started a quest to find who had explained these complex systems.
I hope you're getting this. I'm sure you are. This is a very salient issue he's raising. And it's much more complex than most people would realize listening to him. The basic idea is, in order to prove evolution, you would have to show the pathway of every single mutation. And it could be hundreds of thousands of mutations just to make one tiny change in the organism. Are y'all with me on that idea? And you would have to show how each of those mutations could take place or did take place in a random manner and made an organism, watch this, one mutation at a time, be able to survive better than before. And now listen to what he says when he went to look for the data that would make the theory valid.
In Darwinian terms. And I was astonished that nobody had. Nobody even really tried. More often than not, the most common comment you would see is, isn't it wonderful what evolution has done for us?
So what happened when you read this book? What was the next thing that started germinating?
Well, I got mad. I got angry because
At your at the system?
at the system, yeah. And it wasn't even the 60s, it was the 80s, but I was mad at the system because I had been led to believe something, not because there was this overwhelming evidence for it, but because of sociological reasons. That's what you're supposed to believe if you're a scientist these days. I was angry and I wanted to tell people about it. So the next week I'd walk down the hall at my university and see a biochem grad student and grab him by the collar and start saying, hey, did you ever think of this, that? And he'd shake his head and listen politely, and then when I stopped, he'd say thank you, and he'd turn and walk away. And I did the same with some of my colleagues, and they did the same thing, and I began to see a pattern, that I, was just kind of saying rude things about evolution, but wasn't being very effective. And that went on for a few years, and never crossed my mind to write a book about this. Even though Denton, he was a geneticist, scientist, but he hadn't covered the area of biochemistry. I'm a biochemist. And biochemistry studies the molecular basis of life, the nuts and bolts, the foundation of life. And I saw a lot of things in my own field that he had not mentioned, and but nonetheless, I didn't think to write a book, because scientists simply don't write books. They write technical papers.
Yes, exactly. And in fact, what you were seeing was it not really even another level below in terms of the complexity of what was going on there?
Yes, he discussed mostly organs and homology, which is the similarity between different groups of animals. But these are at the organ level.
Not at the molecular level.
Not at the molecular level.
And that was your world.
Yeah, that's right. And it's interesting because it's also something that Darwin knew absolutely nothing about. Molecules in the middle of the 19th century were theoretical entities. Atoms were theoretical entities. Nobody was sure if they existed or not. And the cell kind of looked like a little blob of protoplasm, they called it. And so nobody thought it would be hard to get a cell. Well, how hard can it be get
a blob?
A blob, yeah. And there's this wonderful story of a guy named Thomas Huxley, who was a friend of Darwin's, and another fellow named Ernst Haeckel, who came upon some mud that was dredged up by an exploring ship. And there was the days of great exploration of the planet. And they looked at the mud under their microscope, and they thought it looked like a cell. And so they thought that cells could spontaneously bubble up from the sea bottom. And they named the cell Bathybius haeckelii, kind of after Haeckel.
The two of them, yeah.
But since then, we've learned that it's a little bit more than just a bit of jelly. And it's much more like a miniaturized factory. Think of something like an automobile or a computer factory, except maybe 100,000 times bigger, filled with all sorts of things, and it's unmanned. The little robots go along, and they have to recognize where they're supposed to turn on a lane and go down and deposit some supplies for another machine to take those supplies and use them to start to build some other machine, which it passes off to something else.
And it's not as though they have little minds. They're just molecules, just assemblies of atoms, and they do these things.
That's right. And and like the nuts and bolts in cars, they have to have the right shape.
Critical to everything.
And the right chemistry, which comes a lot from their shape, or else they don't work.
Yeah. What finally turned the page in you thinking to yourself, I've got to do more than a paper, I've got to write a book?
It was when I met a man named Phil Johnson.
Yes.
Phil Johnson.
An attorney.
Yes. He was a law professor at the University of California at Berkeley. And he too had read Michael Denton's book, and it had impressed him as much as it impressed me. And he read it while he was on sabbatical in London, and by chance he saw in the same store window a copy of Denton's book and a copy of Richard Dawkins' The Blind Watchmaker. And he read them side by side, and he was fascinated because here are two, agnostic or secular scientists starting with the same data and saying that this theory based on the data is, completely wrong, completely right. So he realized that there was something other than just the straight data that was going on.
Something's going on here.
And so he wrote a book called Darwin on Trial, where he said that the major plank supporting Darwin's theory is that science has adopted a philosophy named materialism. And that is that only matter and energy and forces are allowed to be considered.
This was an a priori assumption, in other words, that he was saying they had made.
That's correct. And then before you go in, before you find out what's inside the cell, you say whatever is inside the cell, we're going to explain it by forces and particles and motion and so on. And given that assumption, then something like Darwinism has to be true, because there's no
other option.
There's no other option.
Yeah.
There's no, if you think that intelligence was involved in guiding life or making life, that's a no-no, that's out of bounds. And if you want to be a scientist, you can't say that, because that's not science by definition. So I read Philip Johnson's book, and I liked it. Some people say, a lawyer writing
what
about
does he know?
Evolution. But he makes the wonderful point, which is completely valid, that evolution cuts across many disciplines, and there is no scientist who is an expert in all of the disciplines that evolution covers. So it's much better evaluated by somebody who's trained in evaluating evidence and assumptions, and he put himself forward as that fellow.
Person, yeah.
So I read his book and liked it, and about a week later, kind of serendipitously, I was walking by the department office, and they get subscribed to Science and Nature and
the journals.
And I picked up the latest copy of Science magazine and saw in the table of contents that they had a little short news piece on Philip Johnson's book, and I was, excited because I had liked it. I wanted to see what they had to say, and essentially it was a warning: keep your students away from this book. It's going to confuse them, and worse, it's written by a respectable source, a law professor at the University of California, and that would give credence to.
And wasn't he from Berkeley?
Yes, yeah, Berkeley, yeah. University of California, Berkeley, yeah. Very prestigious.
Not only that, considered it as liberal or free-thinking a place as you could find, maybe?
Yeah, that's right. He always said that he never got in trouble, Philip Johnson didn't, by espousing intelligent design, because he knew that nobody was afraid that Berkeley would be mistaken for a religious institution. So I got mad at the news story too. I wrote a letter to the editor of Science about this, saying, I have read this book, seems pretty interesting. Here's this very smart person with some good questions. It would be better to address the questions rather than be
dismiss it.
Well, not only dismiss it, be condescending to it.
Yes,
they were very condescending.
Yeah.
And they printed my letter.
Oh, they did.
And Philip Johnson saw the letter, and he wrote to me then. And I didn't know it at the time, but I was then plugged into his network of useful contacts. And later on, he invited me to meetings of like-minded folks. And there I got to see philosophers and historians and other people who not only read books, but they wrote books too. And that's where I started to get the idea, maybe I could write a book. And so at one of those meetings, I decided that I had something to say that nobody else had said, and that I was going to try to write a book too. And 2 years later, that was Darwin's Black Box.
And Darwin's black box was the cell. We don't know how it works inside. And so that's why you chose that metaphor.
Yes.
That to Darwin, it was a black box.
To Darwin, the cell was a black box. It was, and the definition of a black box in science is some system or something that does something neat, really interesting, but you don't know how.
Yeah.
It's because it's inside the black box. You
Yeah.
can't see inside. You don't know what it's doing. And generally, you assume it's simple because, it's easier than trying to figure it out. But Darwin and his contemporaries thought the cell was simple. And now we have opened Darwin's black box. Science has progressed enough that we know what's inside it, we know what runs it, and much to the consternation and surprise of science, nobody was expecting this, there are machines, literally machines made out of molecules, like little trucks and little buses and little outboard motors.
Traffic directors.
Yeah, traffic directors, little signals telling a molecular truck, which actually drives along a molecular highway.
Yes, yes.
Say turn left or turn right and
deliver this at a certain address.
That's right. And if you don't deliver it at the right address, you're in trouble. And there are many genetic diseases that result from delivering cargo at the wrong address. Most DNA in the cells of big animals like us doesn't actually code for proteins, which are the little nuts and bolts of molecular machines. For the longest time, people didn't know what it did.
What did he just say? Most DNA what? Doesn't code for protein. You know all about that, don't you?
Many times in the history of science, if you don't know what something
It's
does,
junk.
you say it's junk, it doesn't do
Well, that
anything.
was crazy, wasn't it?
And but just like all those other times in the history of science, we're finding out it isn't junk, and a number of functions have been found in it. It's involved in regulating when, where, and how much the proteins
Are expressed.
How much are made, and there's other pieces called microRNAs, classes of molecules that we didn't even suspect were there 15 years ago, which we now know are critical for regulating genes. It used to be thought that a gene was a segment in a much longer piece of DNA, and that some machinery would come along and make a copy of the DNA called RNA, and then the RNA would be used to make a protein. But it was all nice and neat. It would start here, it would stop here. But now we know that it starts way out there sometimes, and it continues way down there, and that depending on different conditions, you can make a different protein using the same gene. One gene in the immune system of some invertebrates, where it's one gene and you can make close to 40,000 different proteins using that one gene. Yes, one thing you can say is that it's not going to get simpler. Because the complexity we have discovered is there. It's kind of like, again, say a car. You say, look, 4 wheels, and this thing here is a windshield. It helps, protect your eyes, and there's a steering wheel. Wow, but maybe if we dig further, we'll understand that it's not as hard to make a car as you'd think. And then you open the hood, and you see the
Something's
engine.
turning in there.
And it's this completely new level of complexity that you hadn't seen from the outside. That's what we're doing here, and so, all the complexity we've learned so far, and it's continually increasing, is here to stay. And if the pattern continues, it's not going to stop for a long, long time. When I started to write a book, I knew there was a big problem for Darwinism, because Darwin's is a specific theory of evolution. It says change happens randomly, and it happens in small steps, and if it happens to improve something a little bit, then natural selection can come along
and hold on to that.
Hold on to that, favor that, the offspring of an organism would survive better, and so on. But in biochemistry, you learn that there are a lot of systems that don't look like they could be put together just a
step at a time.
Teeny step at a time. It's much more like you have to have several parts together and working before it works at all. And in my book, I gave the example of, in our everyday world, of a mousetrap, an ordinary mechanical mousetrap. A mousetrap has a number of parts. It's got a wooden platform, and it's got a spring, and it's got this thing called the hammer that you push over, and there's a bar which has to stabilize the hammer until the mouse comes along. And I made the point that, you know, the trap needs all these parts to work, and if the spring is missing and the hammer is missing, then you don't have a trap that works, you know, less well than a complete trap. You have a broken trap. It doesn't work at all. And that's what I meant by irreducibly complex. It has a number of parts, and all the parts work on each other to perform the function.
And if any one of them was missing, the organism would have no benefit because the rest weren't doing anything.
That's correct. That's correct. And if you think to yourself, well, suppose I wanted to evolve by Darwinian means something like a mousetrap, by something like Darwinian means. You know, how would you do it? You know, would you start with just a wooden platform and say to yourself, well, maybe I can, you know, catch mice inefficiently by tripping them on the platform as they try to run across. And then maybe you can add what's called the holding bar. It's just this long bar which stabilizes the hammer, because they might trip and impale themselves on the
holding bar.
But, you know, everybody knows that that wouldn't work. So it was a good illustration for me, because it gets across the problem for Darwin's theory pretty easily. If you think that even a little mousetrap has to have a number of parts, these things that are, you know, chemical factories, solar factories, trucks, and so on in the cell, they've got lots of parts too. And so the same problem crops up for a gradualistic, undirected theory like Darwin's. You know, it's exceedingly unlikely to get something to build up gradually. We should say that Darwinian evolution can do some things, and I made that clear in Darwin's Black Box and my newer book too. So you shouldn't, you know, just wave the whole thing away. And some of the things it does are medically and well, they're important, and they're economically important. Things like antibiotic resistance can be explained pretty easily by Darwin's theory. Many genetic diseases can be, the resistance to some diseases, like to malaria.
Early people in Africa who are resistant to malaria.
Yeah, and they have something called the sickle cell gene. And if you have one copy of the gene for sickle cell hemoglobin, then you have resistance to malaria. But as most people know, if you have two copies, one from your mother and one from your father, that leads to sickle cell disease, and that's a deadly disease, as deadly as malaria is. So it's not a real elegant solution to the problem of malaria. And actually, when you see Darwin's process at work, it's frequently, you know, it's virtually always like that. And that is as it is in the sickle cell case. That is, it changes either a tiny thing which will mess things up, because the hemoglobin in sickle cell disease will.
One amino acid is all. Is that right?
That's correct. That's correct.
One letter of the alphabet in the wrong place in a string of several hundred?
Yes, that's correct. And it causes the nice hemoglobin molecules which float around in a red blood cell, that causes them to aggregate into a gel. And in people with this disease, their red blood cells go from very flexible and elastic, like they should be, to something like rocks. They are very inflexible, and that stops them from going through capillaries. The capillaries jam up, and the tissues can't get oxygen and die, and so that's what causes the disease. So Darwin's mechanism works great when it would help you to break something. It doesn't work nearly so well when...
To put it back together.
To put
it together at all.
anything together, yeah. It looks like, this is all of a piece, that the properties of the universe and the utter complexity of life, it was all meant for life, to produce life.
Well, was that fun? Let's take a minute before we leave. I'll comment on these two that I left, but before I do that, would you like to ask a question about anything he was saying that you thought, let's see, what did that mean, or etc.?
I just question when you say that there's mutations, about maybe 3,000. How many of those are beneficial?
Did you hear the question? What about these accidental mutations? Are any ever beneficial? And the answer is almost never. Yeah, in fact, David Berlinski, this very bright fellow that I really want you to hear a little bit from, I heard him say one time, we have a devil of a challenge finding any mutation that would make something better. Just amazing. Really amazing.
So out of... It sounds like there are a lot of mutations that occur regularly that are identifiable, that we've seen in the past. For something to be progressing, you would need to have a mutation that's brand new that we've never seen before.
Yes, it's an interesting world there because these mutations are taking place because of copy error, because of radiation, because of toxins that we eat, sometimes because of toxins in the environment, and all of us have a sense that some of our illnesses are because of that. Probably we blame too much of it on those kinds of things instead of blaming it on the way we choose to live. That happens to be pretty well able to be shown. But your question as to whether it has to be a new mutation to bring some benefit, or let's say there was a Darwinian mechanism operating, it could be a new one or it could be an old one. The Darwinian mechanism says that each mutation is going to make that organism better able to survive. And of course, it looks like that's just outrageously wrong. But I hope you understand, folks, that a person who says, I'm going to be a scientist, and science excludes the possibility of anything but just forces and Newton's laws, if you will. In other words, we rule out any kind of a supernatural or design element. They just have to close their eyes to that if they're going to be a scientist. And so it's an amazing posture, but if I get a chance to play you as much of Berlinski as I would like, maybe 8 or 10 minutes of this, he tells there how, and he's a physicist and a mathematician, both he has a PhD in several fields, and he says the mathematicians just laugh at this. Why would the mathematicians laugh at this idea of naturalistic selection? Because of the improbability of getting it right. And they know that. They just know that inherently. They don't have to sit down and do a calculation. They've been there. And he talks about the physicists. They just shake their heads and say, what are you guys talking about? This is crazy. But the biologists, by and large, have been schooled, like he said. What was evolution taught to them? It was assumed. Isn't that amazing? And when he went to say to himself, now let's see, he couldn't remember learning it. Remember, did you catch him saying that? Very interesting. He goes to the literature. Nothing is there. He said nobody had even tried to do this. Very, very fascinating. Somebody else?
When he was talking about the sickle cell, the sickle cell anemia, it's only in black people.
Oh no, it's true that the black population. It's true that the black population has a much higher incidence. Now you have to understand, and I didn't tell you this ahead of time. I was thinking about it, but since he mentioned it, if you get the gene, if you get the hemoglobin single, and I showed you this, didn't I? The one amino acid that's incorrect. If you got that gene from just one of your parents, we say you're a sickle cell carrier. And that person, it happens by accident that person is more, not totally, but quite immune to malaria. Lots of Africans still die of malaria, and certainly those who have that single gene. But it's correct that people who do not have that single carrier gene, they die if they don't take treatment. And most people that go to Africa to work there as missionaries or whatever, they take prophylactically. If they don't do this, they will die. It's just Western Africa is called the white man's grave. There's just people went over there and died like flies. Missionaries from all churches. But if the person gets it from both mother and dad, then they get this sickle cell disease, which he described. The red blood cell is not flexible. It compromises circulation. And it is a fatal disease, although people live for a few years. They have these crises when it gets real bad and so forth. Anybody else?
If you go and have your blood tested and find out there's no mutation, is there a possibility later on that from maybe what you eat or that you could get?
Good question. By the way, when you go to 23andMe, are you making the connection with that business? They don't look at your whole genome. They pick out a number of places where we know we know of risk, like BRCA1 and 2, for example. And there's about 10 others that we know of now that are tumor suppressor genes and so forth. Normally, it's because we inherit this mutated gene and it's in every cell of our body. It is possible that any one cell in our body from other causes, not because of inherited mutations, could become cancerous from, let's say we're talking about cancer, from these different sources of whether it's radiation or toxins or copy error on that one cell. That's not system-wide. And that one cell could become cancerous. Now, if I had the BRCA1 and 2 intact, and even the other tumor suppressors, even though that cell wanted to become cancerous, they could repair the DNA in that cell and keep it from happening. That's the mechanism that's going on there. So even though here's what's going to happen. I don't know if it's going to be after staying away from wheat fad, but one of these fads, one of these days is going to be get your DNA tested and try to get it fixed. And that's not going to be the answer to our health problems. Now I'm getting into my other world now. I have a graduate degree in nutrition, so that this is another thing I do. And we know that the way people live is huge in comparison to. It's been fairly well demonstrated that all this environmental stuff that we think is such a big issue is probably just several % of our problem. Our problem is the way we choose to live. And that's what my wife and I spend. When we're not doing these, we do those other things and try to help people think of those changes. Somebody else?
I'm not sure how it works.
Just go ahead and try. We'll work together on it.
When you're speaking of mutations, I am gathering the information that we just talked about physical illness, different things that happen. What about the body itself, like people who are born perhaps with one arm or without this part? Are we that mutation?
It can be. When the fetus is developing, you gals all know this, the men may know it too, if you are limited in vitamin K, when the neural tube is forming, what is it, at about, it's only at about 2.5 weeks when that's happening. It's amazing. You get a condition we know as spina bifida. You all know about this. And so every young mother today is told to take vitamin K or eat greens or both as a precaution. So there are multiple issues that go on. And at times it's a genetic issue. You all know about the thalidomide babies. That was a drug in the body that interrupted one of the steps when the cells were copying and planting a limb or something. And there are probably multiple issues there. And we do know that there are, you all have heard this. What's the gene that makes us liable to get diabetes? Have you heard about that? Yeah. People of color, the world over, African or extraction from Africa, American natives, American Indians, Indians from the country of India, people of color, Hispanics, the world over, have a genetic weakness to get type 2 diabetes. And for example, the Pima Indians in this country, who never used to get diabetes because of the way they used to live, today 90% of them get diabetes, type 2 diabetes, in their lifetime. 90%. And if Americans live that way, we only, at this current point in time, 1 out of 14 Americans has type 2 diabetes because they're living like those people live, or they're living like we are. So there's no question that there is a genetic factor for them. And so that's going to be another fad. Connected. I read a long article in the last day or two from a scientist whose name I'll give you in a second if I can think of it, concerned about this. Oh, it was T. Colin Campbell, the author of the China study. In his latest book, have you started reading it yet? The whole book is on that concept, isn't it? What he calls reductionism. We try to think, let me just find this one thing that's my problem. I need to take this vitamin. And his argument, and he's an esteemed scientist, is that the problem is the whole person in terms of the way we've chosen to live. There's not just one thing that's going to fix our problem. Interesting question. Anybody else, just for fun? Something else that he talked about?
Not so much what he talked about, but what we went through with her when she had one of our babies, because I'm O positive and she's
Yeah.
She had to have a RhoGAM shot before the next baby.
Yeah. You had this Rh negative issue. And yeah, kind of a genetic thing, actually, just because of blood types. Anybody else on what Michael B. had to say? All right. Let's just take a moment on these last two. It kind of has been covered by our discussion there together, but how does the mapped genome differ in each person? If you have thought about this, the scientists mapped the human genome. Well, who did they use? Joe Blow? Well, it turns out that our genomes are so similar. There's just a few hundred places where there are differences. And 98% of our genome is the same as what animal, what primate? A chimp. Don't say monkey, say chimp. Just a second here. And so they don't have a problem with mapping the human genome, even though there are variations among people, see. But and so some of you want to know, well, how do they know that this guy was the committer of the crime then? Well, even with just a few hundred or whatever the number is, we can delineate that and figure out whose DNA it was. So is the DNA from my dad and my mom exactly the same? No. There are differences. And it's interesting that in the scheme of the designer, just a few number of genes can make such differences in how long my nose is and whether I have big ears or not, and who knows about what's going on inside, right? Somebody, yes, and then we'll come to Tom.
Last night you said, well, and you just referred to it, that 98% of our DNA is like the chimps.
Yes.
But isn't it true that there are many organisms that are all very similar?
Yes. Yeah, and to the evolutionist, they say, look at the evidence for that. And the creationist or the designer, the believer in design, says, well, the designer is like a person who builds a house. They use shingles, they use windows, they use sticks to make the walls, and they put sheetrock or what, you know. It's common architecture. But it's interesting how much is the same. Although it depends on the organism. There are some, you know, the difference between a dinosaur and human beings is quite a bit different than the chimp and a human being.
I just wanted to comment that we share a lot of DNA with a banana, too.
Yeah, that's right. Yes.
I went to our local college about 10 years ago, and there was a professor from Berkeley that was working with genetics. And he said at that time that we have 99% of the same genetics as a pig. And then he was saying that he was upset because the people in agriculture were using pig genes in strawberries.
Mm-hmm.
He was upset that it's one of the genomes, one of the genes that they're using. And he was upset at that time because he said they had no right to be using this stuff yet until more research was done because they didn't know what that was going to do to the environment.
Yeah, this is the whole, if you could hear what she said, this is the whole world of the GMO question. And I have an hour-and-a-half lecture on that. I know you don't want to hear tonight, so. But it's an interesting observation that you make. You showed us what the genome looks like. How do we know where the gene portion, how does the gene portion look different? You heard, and there's one more area of this that I want you to, I'll take some time tomorrow evening to sort of show you, and maybe some the next evening, but did you catch this? That Behe said that there is one gene we found in an invertebrate, and we are invertebrates, you know that. Or I'm sorry, we're vertebrates, and this was found in an invertebrate, that one gene could make 40,000 different proteins. I'll show you how that can happen. And here's the interesting thing that we did not know until the whole genome was sequenced and it finally came home to what was going on. Scientists thought there should be 100,000, maybe more genes, and we only found 20,000. The answer to that question is what he was talking about. And what do you suppose tells the protein, tells the gene which of those 40,000 different proteins to make? It's the junk DNA. It's the control region, see? And here's the amazing thing. Now here I'm getting into my other territory. The way you live, we know, changes your own DNA. And when you wake up and say, I'm going to live right, somehow it gets fixed. Now what about that? It's an amazing thing. We have hard data for that. It's quite remarkable. And it's only going to get better or more interesting as men and women scientists probe these questions. Well, maybe that's enough for tonight. I'm trying to get you always out of here at 8:30, and I thought a few minutes ago it looked like we had several minutes, and it took 10, so forgive me for that. So if you have any questions, leave them with someone at the door. And we'll see you tomorrow evening. And in tomorrow evening's class, we'll do the interview with Ben Carson.